Good morning, everyone. My name is Sreenidhi Maddineedi, and this is my presentation, Investigating Hedgehog Inhibition in Tanky Rays and Mitigating Wind in Sarcoma. So that was a lot of big words. Let's go ahead and break that down. So first of all, what is sarcoma? So sarcoma is a type of muscle cancer or soft tissue cancer, and there's three main types of them. There's angiosarcoma, there's rhabdomyosarcoma, and leiomyosarcoma, the latter two being of interest in this project. So rhabdomyosarcoma is the most common soft tissue cancer in children, and leiomyosarcoma is a uterine lining cancer that's often seen with a very, very high prognosis, nearly a 14% survival rate in fact, and there's roughly 1,200 cases of it diagnosed yearly in the US. So now talking about what the hedgehog and Wnt pathways are. So hedgehog and Wnt are both cell cycle pathways that are upregulated in sarcoma. Essentially the way it works is that whenever you have hedgehog overexpression, there's sort of this cofactor that goes ahead and increases the Wnt tanky rays pathway inhibition as well. And that's oftentimes what scientists are seeing is that whenever they're trying to target the hedgehog pathway, there's a sort of environmental factor that goes ahead and artificially increases the tanky rays Wnt as well. So what this project seeks to do is sort of artificially inhibit both pathways and seeing what the impact of that is on metastasis and oncogenesis in sarcoma cancers. So that's exactly what the research gap is of the project and what it aims to address. My hypothesis was that if RD and SKUT1, RD being the rhabdomyosarcoma and SKUT1 being the leiomyosarcoma, if they're both treated with GAN61 and XAB939 chemical inhibitors, then the cells would express a reduced cell growth and metastasis, as these cells would now be exhibiting these more healthy and not overexpressed levels of the GLI and Wnt proteins. So talking a little bit more about the cells in the treatment groups. So again, SKUT1 is the human uterine leiomyosarcoma, and RD is the rhabdomyosarcoma. In fact, when we ordered these cells from ATCC, the SKUT1 was roughly at a 75-year age before the cryopreservation, and RD was at roughly a 1-week age, which will be of interest later on in the experiment. And now talking about the treatment groups. So XAB939 is essentially a hedgehog pathway inhibitor, and GAN61 is a Wnt tanky rays pathway inhibitor. And hence the treatment groups, so we're testing the control, which has none of the chemical inhibitors whatsoever. This will be just growing and proliferating as we expect any other sarcoma to. And then there's different treatment groups that contain them at different concentrations in order to test the effect of the cytotoxicity as well on the cell growth. So talking about the different assays or the different treatment groups and how I sort of conducted the experiment. First is the migration assay. So the migration assay is essentially exactly as it sounds. It's meant to show cell growth and movement into new areas, thereby indicative of possible metastasis. And essentially the ways that you would conduct this assay are by first culturing the cells in a 12-well plate and waiting until it reaches 100% confluency, so roughly 3 days for the culture. And then treating the cells with a serum-free media, which means no FBS, no PBS, really just the regular Dubesco-modified serum and such. So after treating the cells with the serum-free media, we essentially incubate them and then treat with the chemicals, again at the different concentrations as according to the treatment group. Then you can make a scratch with a 200 microliter pipette tip, and essentially what it's supposed to measure is that we're going to see how much the cells migrate into that scratch area. So data was then analyzed as a percent change in scratch area as compared to day 0, and I did this over 3 days, so days 0, 1, 2, and 3. Over here actually is one of the SKUT1 treatment groups. So you can see this is sort of where the scratch was on day 0, and then you can see how the cells sort of migrated into the scratch area by day 3. So second was the Alamar Blue assay. Now the Alamar Blue assay was essentially intended to measure the cell metabolism, hence indicative of the metabolic activity. With higher fluorescence, you can also see higher metabolic activity in the cells. So for this, you're basically using a microplate reader. It's very similar to how you sort of start off your migration assay. You first culture your cells, this time in a 96-well plate, just because that's what's most compatible with the microplate reader. And you wait until it reaches 70 to 80% confluency. Now the reason why you need a lower confluency, or you can use a lower confluency level for this compared to the migration assay, is because with so many different treatment groups and so many different wells, it should essentially be able to corroborate the data that we see in the different ones. With migration, the reason that you want it to reach 100% confluency is because you want these cells to be at their peak condition, so you can really measure their activity sort of in the most advanced environment or in the most advanced cases. So then you treat the cells with 100 microliters of the serum-free media and incubate roughly for 24 hours, and then you go ahead and add in 10 microliters of the Alamar blue reagent. And this reagent is essentially what's going to cause that fluorescence level that we'll see in the microplate later. And measured at 750 nanometer absorbance and 600 nanometer absorbance. Again, the reason for that difference is because 600 nanometers is what's compatible with just the media. So if you were to subtract that from the 570 nanometer absorbance and fluorescence levels, is just showing exactly what the chemicals are causing. And the third and final assay is quantitative PCR. So PCR, as some of you guys might be familiar with, stands for polymerase chain reaction. There's several different types of PCR. You have RT-PCR, qPCR, as well as RT-qPCR. Now the reason why I went with qPCR is because this is quantitative and that it gives you actual hard data measurements as compared to something like real-time PCR. So again, the steps are very, very similar. You're just culturing the cells until they reach confluency. Now what's different about PCR is that it's far more involved. You have to first extract the RNA, then use that RNA as a template for your complementary DNA synthesis, and then after preparing this is what you can actually begin to assay with by preparing the qPCR plate with an oyster mix and the custom primers. And that also is a little bit of its own process. So the way that you come up with the primers is that the purpose of PCR is to test the expression level of various genes. So in my experiment, there were 11 genes of interest, GAPDH, SAH, HPRT1, SNO, Glee receptors, and I could go into what all of those are. But essentially, there are 6 genes that are testing hedgehog-specific pathway expression, and then there's going to be 2 more genes that are testing Wnt pathway-specific expression, and there's 3 that are involved in crosstalk. What we're hoping to see is sort of a suppression of all of these 11 genes expression in the different treatment groups to see what's most optimal, again, for sarcoma inhibition. So moving into some of the results, and we're just going to go as sort of that consecutive order of how we talked about our methodology. So starting off with migration, we're going to split this up into just SKUT1 cell lines, and then the RD migration in those cell lines. So essentially what you can see here is that XAB939 significantly decreased migration. And what's important to consider when you're looking at this bar chart is that the y-axis is meant to show the percent of the original scratch that remained after day 3. So you want this to be actually as high as possible because that way it shows that there were no cells that were migrating into this area. And then with the RD cells, we saw that all treatment groups did express some sort of statistically significant decreases in the migration as compared to the control. So we can see in both cases here with the control, opening my laser dot works. Okay. So what we can see in both cases with the control is that it's a very small percentage of the original scratch that was actually remaining. And again, that's just showing that these are 2 sarcomas that are very, very heavy in metastasis and they're very aggressive cancers. So because of this high rate of migration, we can see that with the different treatment groups, it's actually stopping that, it's stopping the oncogenesis from happening. So then moving into Alamar Blue. So what was unfortunate with the Alamar Blue assay, and I think a large part of it was because of the incubation periods. Most of the cells were just incubated for roughly 2 to 3 hours. Perhaps incubating them for 4 to 5 would have caused more statistically significant results. But essentially, there were no significant differences between the treatment groups, both for RD and SKUT1. And it kind of just shows that the XAB939 and GAN61 treatment, both in combination and singularly or independent of one another, do not significantly impact cell metabolism as much as it does growth and proliferation. And then moving into qPCR. So the reason why I went with this sort of expression for the qPCR results is because actually showing you all the charts would just overcomplicate this entire situation. But essentially what we saw with qPCR is, again, sort of independent. So first looking at SKUT1 and then the RD cell groups. With the SKUT1, we saw, again, very, very similar to the migration assay, where XAB939 proved to be more beneficial because this is what really decreased the hedgehog-Wnt pathway expression. However, GAN61 also did decrease expression of both pathways, just to a lesser extent. And then in the RD cell groups, we found that XAB939, again, which is inhibiting the hedgehog pathway, would actually increase the Wnt expression, while GAN61 decreased expression of both pathways. So this shows that GAN61 might have proven more effective in the RD cell groups, whereas it was XAB939, which was more effective in the uterine leiomyosarcoma. So, okay, I don't know what the overlay is there, but essentially talking about, again, just sort of summarizing all of our results, first looking at independent treatment groups. groups. So we saw with the uterine liosarcoma that telomerase inhibition, so via the GAN61, proved to be a very strong treatment group for the aged liosarcomas. And the reason I specified aged liosarcomas is because, remember again, these cells were roughly 75 years of age before the prior preservation when I received them. And it shows that there is this sort of glee or hedgehog inhibition that might actually produce less remarkable effects, whereas with the rhabdomyosarcoma, which was of a younger age, has just as cancer and as a cell line, hedgehog and Wnt inhibition both hindered the early rhabdomyosarcoma progression. So again, sort of what the exact implications of these are is that with main concerns with drug efficacy, the aged sarcomas may actually develop some sort of innate pathway alternative expression. So this is what's going to help it overcome the hedgehog inhibition and actually proliferate to a greater extent. Whereas with combining both the hedgehog and Wnt pathway inhibition, because we're sort of targeting it from both ends, this is what's really going to reduce the cell viability, and again, both in aged and young cell lines. And talking about the sort of threshold effect which I observed in SKUT1 cell lines, it seemed that at some point at a higher concentration, so at a 30 micromolar versus a 10 micromolar concentration, the cells actually began growing back. And it's sort of because there is this inherent level that the cells are designed to recognize whenever there's this level of toxin or such in its environment, that the cells sort of develop this sort of alternative mechanism to actually increase their cell cycle and increase their proliferation. So being able to sort of model this perhaps further down the line in mice models and being able to actually target that cytotoxicity level or target that threshold level would be one of the more optimal ways to sort of draw conclusions from this experiment. But yeah, so with future work, again, potential errors from this experiment might have been with insufficient RNA for the PCR. So with PCR, there's two main things of interest that are with the cell cycle number, which basically shows, so in PCR, essentially the way that I kept in mind is that it's a set of 45 cycles that run. And the way that you see what gene expression there is you look at which had the earliest cycle level of expression. If, for example, our GAN61 a treatment group at a 10 micromolar concentration saw glee expression at, let's say, cycle 10, versus the one at a 10 micromolar concentration saw glee expression at, per se, the 15th cycle, we can see that there was increased glee expression in the GAN61 at the 30 micromolar concentration, just because the system was able to detect it that much earlier. So insufficient RNA in some of the treatment groups might have been responsible for why we saw the sort of like interesting phenomenon that was observed just between trial 1 and trial 2, where in like trial 2 had more results that were corroborative of our actual conclusions. Another thing also was that I attempted to run all my samples in triplicate, so that essentially means conducting 3 trials. But certain RD migration samples were just unusable because they were either actively done in serum-containing media, not serum-free media, or it's just that the image-stage software, which is what I was using to analyze all my results, couldn't actually pick up where the actual scratch area was. So again, further, and moving on from that, talking about additional assays that I could have conducted, one of the things I would love to do is actually maybe do an Elmore blue alternative to again find that migration effect with also coupling it with metabolism. So conducting additional assays to really investigate the crosstalk, and also doing a cell counting assay to really see what the viability number was. So a cell counting assay basically shows you how many cells there are total in your sample, and how many of your cells are actually alive or actually viable. So doing a cell counting assay would have been able to assess whether those changes in metabolic activity were due to varying cell numbers or some sort of other environmental factor or stimuli. And further applications of my research is again in targeting compensatory resistance in sarcoma treatment, as well as identifying specific synergistic inhibitor strategies, again looking at the crosstalk between the hedgehog and Wnt pathways, and also mitigating adverse effects of the aberrant pathway activation while applying inhibitory treatments. So these are some of my references, and acknowledgements for my project. I want to thank my mentor, Mr. Kurt Chang. He was with me in the lab almost until 11 p.m. on most days. So yeah. But with that, thank you so much for listening, and we'll have our next presenter come up shortly.